Dr. Anup Kumar. Welcome to my YouTube channel PharmEase. In this video, I am going to explain you one of the important pharmacokinetic which has of clinical significance, which is called as the drug clearance. Remember, drug clearance is a property of the drug, and as the drug entered into the body, it acquires certain volume in the field of, so usually we express the drug concentration in microgram per ml, and after a period of time we will observe that the drug has been eliminated from the body, which represent, usually it is expressed in the elimination rate microgram per hours or the time units. So basically in pharmacokinetics we need one more parameter which is required, a sense how much volume of the body is free from the drugs. So that is why we need important pharmacokinetic parameter which is called as the drug clearance. So we can define as the drug clearance is the volume of the body or the any specific volume which is get cleared of the drugs in per unit times. So basically, as I have shown here, it is a proportionality constant. Clearance is a proportionality constant between the two important variables. One is the plasma concentration, another one is elimination rate constant. It can also be explained through that clearance is dE by dt divided by the plasma concentration, which is calculated through the pharmacokinetic modelings. The clearance has the unit of volume per unit times, either it is a liter per hours or the ml per minutes. Second important parameter which is related with the clearance is the elimination rate constant. Say here is the elimination rate, here is the elimination rate constant into the volume of distributions. So it is the basically the proportionality volumes, or we can say that KEL, and we combine together we will get that how much quantity of the volume is get cleared. Another way to express the clearance is using the non-compartment modeling, is basically the ratio of the bioavailable dose divided by the AUC 0 to infinite. It is calculated by non-compartment modelings. So how does we calculate the AUC? I have explained, I have made a video which is given. Physiological modeling, we can say that the clearance is the amount of, it is a product of extraction ratios and the blood flow rate or perfusion rate to that organs. In the physiological modelings, the quantity of the drug entered into the concentration of the drug entered into the organ minus the concentration eliminating from the organs, which is a proportionality ratio. If the extraction ratio is more than 0.7, we can say the clearance is obviously high. There are the different organ clearances. We can say that the total systemic clearances is the quantity of the drug which is cleared, quantity or which is cleared from the multiple organs like lung, renal or the livers. Also on number of different organs are there, so we can say that the total clearance. So in each cases, how we will measure the clearance, it measured by extraction ratios multiplied by the blood flow rate or perfusion rates. So for example, if the drug is having the clearance of 15 ml per minute, and if the drug is having the total volume of distribution is 12 liters, so how much time is the total volume is get cleared? So usually it is very simple that the how much quantity of this get cleared. So we get to know that 15 ml cleared in a per minute, and we will multiply, and by ratio proportions we can calculate that around 800 minutes is sufficient by which the drug has been eliminated. Since the drug has been the eliminations rate and the plasma concentration varies in the bodies in the dynamic state, since the drug is following first order kinetics, so this is not always the same. When the drug concentration is get changed, so elimination rate is also changed, but in the first order kinetics the clearance remain the same. Clearance is fixed. So as I've taken here the example B, if the drug clearance is 15 ml and the plasma concentration is 2 microgram per ml, then the elimination rate is 30. If we are increasing to 5 times, so the CP is increased to the 5 times, here the plasma concentration increased to the 10 micrograms, so obviously the elimination rate is also increased to the 5 times, but the clearance remain the same. So how we measure the clearance? Clearance is measured usually by means of the compartment modelings, by means of the non-compartment modeling, and by means of the physiological modelings, in which we can find out by means of the zero order or the first order kinetics, or we can calculate by means of the IV infusions methodology. We can calculate by means of the AUC by dose relationships. Important significances of the clearance is usually, since it is one of the important pharmacokinetic parameter which has the clinical significances, since it is representing the how the drug is eliminating. So one way is to express elimination of a drug is through the rate of elimination, or the elimination rate constants, but simultaneously we can explain the elimination by means of the clearance. Clearance is a clinical significant parameters beside pharmacokinetics parameters. Since clearance is a proportionality constant, we can say we can adjust the dose size and we can adjust the hepatic or renal dose adjustment in case of the. Hope you find this video on clearance informative. So please like, subscribe and comment on this video. Thank you.